Dear all participants of the 6th International Conference on Food and Applied Bioscience 2022, my name is Atta Aditya Wardana from Kyushu University, Japan, as well as Bina Nusantara University, Indonesia. On behalf of the authors, I would like to present our research poster entitled Antifungal Performances and Characteristics of Alginate Lemongrass Oil Pickering Emulsion for Fruit Coating. A penicillium species is one of the most harmful phytopathogenic fungi in the agricultural sector, mostly found in citrus fruit. Antifungal coating based on Pickering emulsion from sodium alginate combined with lemongrass essential oil, which contain bioactive compound of geraniol and nerol, have been considered as a promising strategy to overcome it. The use of nanocellulose have been gaining interest due to their prospective features, including hydrophobicity and high adsorption capacity. Then the objective of this work are to develop an emulsified coating film for formulation based on alginate and lemongrass essential oil stabilized with nanocellulose Pickering emulsion, and to study the antifungal features against penicillium species and the coating film properties. The stabilized Pickering emulsion agent stock was prepared by dispersing cellulose nanofiber into distilled water using high-speed homogenizer for 1 minute. At the same time, alginate solution was prepared by gelatinizing sodium alginate powder. Then all the materials were homogenized at 15,000 RPM for 2 minutes. As seen in figure 2b, cellulose appear in typical rod-like shape and form some cluster. To know the interaction of each materials, the coating films was analyzed by using FTIR using ATR method. As seen in figure 2c, there were decreased trend of intensity when emulsion were inserted. It indicated that the presence of hydrogen bonding interaction between the alginate and Pickering emulsion may disrupt the molecular networking between individual beta-D manuronic acid and alpha-L-glucuronic acid. And the greater intensity appeared for higher level of oil, indicating to the enhancement of hydrogen bonding interaction between the component and hydroxyl group from essential oil and cellulose. The microstructure of each coating sample was observed by using CLSM, shown in figure 2D. Obviously, the emulsion droplets was uniformly distributed in the photograph, indicated with yellow color, and displayed a spherical shape. And moving on the results, Penicillium digitatum and Italicum species are considered as the major cause of fungal decay found in the fresh fruit commodities. Hence, in this work, the inhibition of spore germination and survival were investigated. Visually, as seen in figure 3c, Penicillium italicum spores were more susceptible in comparison with those of Penicillium digitatum. Statistically, compared to control and alginate-treated spores, the antifungal features of alginate essential oil increased dramatically, and the inhibitory effect continuously enhanced by 88.28% for Penicillium digitatum and 91.94% for Penicillium italicum when higher essential oil of 0.75% was augmented. Then propidium iodide is well known as a fluorescent dye that is able to penetrate the dead cells. As seen in figure 3, the result implied that the membrane integrity was impaired by lemongrass essential oil loaded coating treatment, leading to metabolic disruption and the death of fungi. To know roughness properties, AFN method was selected. As seen in figure 4, reduction pattern of smoothness demonstrated in Pickering emulsion incorporated coating were corresponded to the occurrence of oil aggregation or creaming phenomena as consequence of evaporation step during thin film forming. And it could be observed visually by 2D topography and line profile in which repeat aggregation were seen forming upland structure. Result also revealed that the incorporation of emulsion resulted in significantly decline in hydrophobicity measured as water contact angle, shown in figure 5A and B. SEM analysis revealed that alginate thin coating film exhibited a compact, continuous, and homogeneous appearance, and no clear separation was found. However, heterogeneous interior micrograph was displayed after inserting emulsion, and more amorphous structure easily observed at higher proportion of oil. Finally, the conclusion of this study, the incorporation of essential oil cellulose Pickering emulsion into alginate coating was found to improve the performance of antifungal activity, and also it had a potential to improve the functional properties such as water contact angle and surface properties. Thank you.